First speaker today is Ashleigh Burke, who I think joined my lab in 2016. So she was actually my very first PhD student. I think hopefully you're about a week away from submitting your thesis, Ashleigh. So yeah, but it's been a real pleasure to have Ashleigh as one of your first students. Is like obviously was a great pleasure. She's worked not only very hard but also incredibly effectively during the 3 to 4 years that she's been with me. I think following her PhD, she's going to go and do some work with you and your Merck, which would be a great experience for her as well. So yeah, I don't want to say too much more. I'll just pass over to Ashleigh, who's going to tell us about her research.
Hi everyone, I'm Ashleigh Burke and I'm a final year student in Anthony Green's lab. So today I'm going to talk about my PhD project, which was titled Design and Evolution of an Enzyme with a Non-Canonical Organocatalytic Mechanism. So just by way of introduction, I'd just like to recap the approaches that were discussed last week and at the start of today's meeting about different approaches to engineering enzymes with new functions. So one of the most widely used methods is to repurpose nature's enzymes to carry out synthetically useful reactions. So we heard again last week about the engineering of a transaminase to produce sitagliptin. And so that's one of the most widely used methods today. However, we're not really changing the intrinsic mechanism of the enzyme. We can also exploit the mechanistic promiscuity of enzymes as a gateway to proteins with new functions. And this has been highlighted recently by the Arnold group, and they've re-engineered P450s for new to nature reactions. There's also the computational protein design approach, which is in its infancy, but it has the potential to be highly versatile because we can think of designing enzymes for any given reaction. So this works by using a computationally designed transition state that's then stabilized by amino acid residues and then docked into proteins in the protein data bank. Any promising designs are then tested experimentally, and their activities are improved through rounds of directed evolution. So we've seen examples published for a small set of well-characterized reactions that have all had their activities extensively improved through evolution. And we saw last week how we're starting to expand this to incorporate more mechanistically complex and energetically demanding transformations. So all these approaches are limited to nature's 20 canonical amino acids. And from a chemist's perspective, this really restricts the amount of chemistry that you could imagine taking place in an enzyme active site. So one of the things that we're interested in the green group is to install non-canonical amino acids that are inspired by small molecule catalysts. We encode these directly through the translation machinery to hopefully create new enzymes with new functions that aren't seen in nature. And as these non-canonical amino acids are incorporated directly using the cellular translation machinery, they should be amenable to directed evolution methods that we use in the lab today. So I'll just give a brief overview of how we do this for anyone who's unfamiliar. So we use a technique that was pioneered by the Schultz laboratory, which allows site-selective and covalent incorporation of a chosen non-canonical amino acid. And we do this by using an orthogonal aminoacyl tRNA synthetase and tRNA pair. So your synthetase attaches your non-canonical amino acid to the tRNA, and this is then delivered to the ribosome in response to a unique codon, and translates your protein containing your non-canonical amino acid. So this strategy has really 3 basic requirements. So first of all, you need a non-canonical amino acid which can enter the cell and is stable to metabolism. You need a unique codon that can be recognized by the anticodon on your tRNA. And you need an orthogonal tRNA and synthetase pair. So to date, there are over 200 distinct non-canonical amino acids that we can install into proteins, incorporating a wide range of functional groups. We use the TAG stop codon, and we repurpose that for our unique codon for our orthogonal tRNA. And for the orthogonal tRNA and amino acyl tRNA synthetase pair, we use the highly versatile pyrrolysal system that's taken from an archaic bacteria. So the tRNA doesn't generally need that much optimization. So this strategy really boils down to engineering your orthogonal synthetase to accept your new amino acid. Okay, so I wanted to use this strategy to design a functional esterase that was capable of hydrolyzing activated esters with high turnover. So in the literature, there's been many attempts to design functional esterases using a wide variety of techniques. So we've seen histidine nucleophiles being installed into protein scaffolds, and the computational design of catalytic dyads and triads, including cysteine and serine nucleophiles, and also small synthetic peptide mimics as well. However, all these previous attempts have been limited by a slow hydrolytic turnover. So if we look at the reaction mechanism, we can see it's split into two parts. So the first part of the reaction is the nucleophilic attack by the catalyst to release the first product, and that forms this acyl enzyme intermediate. And we then have hydrolysis by water to regenerate the catalyst and give the final product. So the first step, it generally works fine, and we form this acyl enzyme intermediate, but all the previous design attempts seem to get stuck at this intermediate and have really slow hydrolytic phases. So far we've not been able to replicate the sophisticated catalytic machinery that we see in nature's enzymes that allows activation of this intermediate or activation of water towards hydrolysis. So in order to design my esterase, I needed to choose a protein scaffold. So I chose this BH32 scaffold, which was a computationally designed scaffold from David Baker's lab in Washington. And it was originally designed to perform this Morita-Bayliss-Hillman carbon-carbon bond forming reaction using a histidine nucleophile. So I found that BH32 was a promiscuous esterase, and it can hydrolyze these fluorescein esters. So the first step releases the fluorescein, which we can monitor on a UV-vis spec at 500 nanometers, forming this intermediate and then hydrolysis to the final product. And we can see here the reaction profile of BH32. So we've got the first step of fast nucleophilic attack releasing fluorescein corresponding to a single turnover. And then we've got this slow hydrolytic phase. And this biphasic behavior is the problem that's been seen in all designed esterases before. If we replace the histidine with alanine, we can see that we abolish the first step in activity and we just have the background hydrolysis. So that confirms that histidine is our nucleophile. So I wanted to have a look into some further mechanistic detail, and I inhibited BH32 with this mechanistic inhibitor bromocetaphenone. And we can see from the crystal structure that the histidine is reacting through this N-epsilon nitrogen. So it's forming this specific intermediate here. And we know from small molecule catalysis that if this intermediate is protonated, that the hydrolysis phase is much quicker. So we hypothesized that if we could methylate at this delta position, we could form a positively charged intermediate controlling the charged state of the intermediate, which would be much more amenable to hydrolysis. So I replaced the histidine with the non-canonical methylhistidine, creating a new enzyme which we called OE1. And you can see here the activity profile. We've now got a nice linear profile, and we've increased the hydrolytic phase of the reaction. However, we have lost a bit of the initial activity. So we wanted to see if we could really improve this through rounds of directed evolution. So I started with the variant OE1. And the first round of evolution, I did some casting libraries and installed 3 extra mutations into the backbone. The second round of evolution, I did random mutagenesis and saturation mutagenesis, installing another 2 mutations into the backbone. And the final round of evolution, I did saturation mutagenesis and combined the beneficial diversity and installed a further 2 mutations into the protein. So we can see that through each round of evolution, we've increased the hydrolytic activity of our enzyme, and we've still got this nice linear reaction profile. So one of the important mutations we installed came in the last round, and that was this L10P mutation. And it actually gave us a 3-fold increase in Kcat as a single point mutation. And I next did some further kinetic characterization. So here's just a mutational map. So you can see all the mutations installed were around this methyl histidine and around the active site of the protein. And here are the Michaelis-Menten kinetics. So overall, we had a 15-fold increase in catalytic efficiency, and this was dominated by a 10-fold increase in Kcat. We then had to do the scary experiment of installing histidine back into the protein to check we hadn't switched on any activity there. And we can see that with histidine, the activity is completely abolished. So the hydrolytic activity is completely dependent on our non-canonical nucleophile. And we also see that OE 1.3 is 9,000-fold more efficient compared to the free methyl histidine amino acid in solution. So this highlights the benefits of installing these small molecule organic catalysts into tunable protein scaffolds. To understand the origins of this improved activity, we then looked at some structural characterization. So here we can see an overlay of our starting variant and our final variant. And we can see there's not much change in the overall global structure of the protein. Most of the changes have come around the methyl histidine in the active site. So if we zoom into that area, we can see that this asparagine mutation that emerged in the second round of evolution is now coordinating 2 ordered water molecules in the active site. So this is creating a network of polar interactions that could be useful for proton transfers or in the hydrolysis, second step of the reaction. And I also want to highlight, again, this L10P mutation. So you might not be able to see it very clearly on here, but in OE1 in blue, this is a completely disordered loop in the structure. Whereas in OE 1.3, we start to see some alpha helices character developing at the site of this mutation. So we've made a change to the backbone of the protein with the installation of this mutation. I then inhibited our final variant with the mechanistic inhibitor bromocetophenone, and we saw site-selective inhibition at the methyl histidine, confirming its role as a nucleophile. And interestingly, we also saw this 9-degree domain opening in the protein. So the protein can be divided into 2 domains, the cap domain and the core domain. And upon inhibition, these 2 domains actually open by 9 degrees. So they still, individual domains still overlay very well with each other. They've just moved further apart. So this is really interesting because if we look at the original protein structure, it comes from the haloacid dehalogenase family. So haloacid dehalogenases have their active site built into the core domain, whereas we started building ours into the cap domain, but they also have these domain motions associated with their reaction coordinates. So you could say we've reintroduced some of the original protein's features through our evolution. And we know that this is a feature that's emerged during evolution because when we inhibit our previous variant, OE 1.2, we still see the site-selective inhibition, but we don't see this domain opening between the 2 domains of the protein. I then went on to do a substrate profile of our variants. So I synthesized a range of fluorescein esters and evaluated the activity towards each one. We can see that they're all well tolerated, and there's been an increase in activity to each substrate through evolution. I was then interested in our variants. So I designed this substrate which has an alpha methyl group. So this is a motif that's seen in some drug molecules. But unfortunately, you can see that the activity was diminished, and there wasn't much of a preference for either enantiomer. So this led me to want to do a further round of evolution. So I started with OE 1.3 and I picked 20 positions, focusing on this hinge region in the protein and around the active site. And you can see here that after the installation of 3 new mutations, we've now got another 15-fold increase in activity towards a single enantiomer. And we've now got an 8-to-1 preference towards the R enantiomer compared to about 1.5-to-1 in the previous variant. So these 3 new mutations we can see on this model here. These are actually all in the hinge region of the protein, and sort of focused around this proline 10 mutation that we installed in the last round. So again, this highlights the importance of this hinge region in the protein and its relation to activity. So far I've shown you that methyl histidine can be considered a genetically encodable surrogate of small molecule catalysts. And I've used this to overcome a long-standing challenge in designing hydrolytic enzymes. And we've also seen that genetic code expansion can be combined with directed evolution to provide a new strategy for creating enzymes with new functions. So now I'd just like to give you a snapshot of some of the work that we've been doing since this work was published last year. So first of all, we're looking at expanding the repertoire of our methyl histidine containing proteins. So we want to try a more complex and energetically demanding transformation, so this Morita-Baylis-Hillman transformation. So one of the students in our labs working on this, so first of all, she took the starting BH32 template and installed methyl histidine, and unfortunately it was inactive towards this transformation. So we heard last week how this BH32 protein is being evolved with histidine towards this reaction. So she chose a selection of variants from that evolution project and found that the round 6 variant with methyl histidine incorporated actually shows some activity now towards this Baylis-Hillman reaction. So she started her evolution project from that starting point, and so far she's completed 1 round of evolution. And we can see here from this HPLC trace that she's already got a really good increase in activity from just 1 round of evolution. Secondly, I've been working in collaboration with a lab in Paris, so Andrew Griffith's lab, on developing a microfluidic assay for this esterase enzyme. So some non-canonical amino acids can be quite hard to synthesize or procure. And microfluidics has the advantage of being on a miniaturized scale with ultra high throughput capacities, allowing you to use sub-milligram quantities of your non-canonical amino acid. So microfluidics it works in an emulsion of picoliter aqueous droplets in a bulk oil phase, and each droplet is an individual reaction containing all your reaction components. So we have a single cell containing a single enzyme variant, in our case also with our translation components and our substrate and product. So we carry out the experiment on a chip like this. So we have the oil phase flowing through this way, and then we have our aqueous inlets injecting our cells, substrate and lysis buffer. And then as the droplets travel through the delay line, the reaction takes place, and they come to this sorting module. So our droplets are sorted based on the fluorescence of the fluorescein product. So we set a threshold, and then any droplets that have higher fluorescence will go one way, and any droplets that fluorescence is lower than the threshold, they'll go in the opposite direction. So far I've used this assay to carry out a single round of sorting on an error-prone library. So this graph shows 90 random clones chosen before sorting and 90 clones after sorting. And we can see we've completely changed the activity distribution of the library. So before sorting, we had only about 5% of clones that had similar or greater activity than our starting variant. And after just one round of sorting, we're now up to about 45 to 50% of clones. And you can see how the activity distribution's changed in the enriched library. And I just want to highlight as well the scale of this experiment. So here I've screened about 4 million droplets at a rate of about 1,000 droplets per second. So microfluidics can also give you some really good information about sequence activity relationships. So you can sequence all the clones that come out of either end of the sorting, and also hopefully pick out some really active clones as well. And finally, I've actually been looking into the translation components themselves. So I mentioned earlier we use this pyrrolysal system that's highly versatile. Specifically, it's this MASI system. So the MASI synthetase has 5 mutations introduced which allow it to incorporate methyl histidine. However, the MASI synthetase is actually really difficult to express and purify and get structural data back from. So it's a 2-domained protein, and most of the crystal structures are of the individual domains, and they have some portions unresolved. So Jason Chin's lab have recently published a mutually orthogonal pyrrolysal alvus system, which has only a single domained synthetase, which is much easier to get structural information from. So I've been able to show that we can transplant these 5 mutations from the MASI system into the new alvus system and use this to produce our full-length OE 1.4 protein containing our non-natural amino acid. And I've also been able to purify this synthetase, and I've put crystal trays down containing the relevant cofactors. So hopefully when we can get back in the lab, I'll be able to find some nice crystals waiting for me and send them off to the beam. So this will really help give us some good structural data to help us evolve future orthogonal synthetases to be able to incorporate new non-natural amino acids that we're interested in. And also I mentioned that these 2 systems are mutually orthogonal. So that means they can work together in a single protein to incorporate 2 different amino acids. So you could imagine using the alvus system now to incorporate our methyl histidine, and this then frees up the MASI system to incorporate a new amino acid. So we can think of designing more complex active sites containing more than one non-canonical amino acid. And so that's all I want to talk about today. So I'd just like to thank my supervisor, Anthony Green, for the opportunity to work on this great PhD project, and Sarah, who I worked on the Esterase project with. Rebecca and Amy are the students carrying out the Baylis-Hillman evolution projects. And we also have our MIB collaborators and collaborators in Paris. Thank you everyone for listening, and thank you to Anya and Alison for organising this conference, and I'm happy to take any questions.
Thank you very much, Ashleigh, for the presentation. We have time for a couple of questions, but I would like to suggest that we use the hand raise feature for the questions, because we have 136 participants, and so I think it would be much easier if we use the hand raise feature. If you don't know how to get that, if you click at the bottom of your Zoom screen on the participants list, then this will pop up, and that has a hand raise feature in it. we'll just have to quickly figure out if you raise your hand, only the host, so Anya can see that, or if we all can see that. Oh, okay.
Can you see me raising hands?
Yes, I can.
Okay, actually, really great talk, and yeah, the microfluidic system seems to be ideally paired with non-canonical amino acid incorporation. What do you think are the barriers to switch to the in vitro transcription translation systems in order to execute evolution this way?
Yep, so some of the barriers that we think so far are obviously using the orthogonal translation components in the IVTT system. So that's potentially one of the benefits of using this new ALVUS system, that we can easily purify out the synthetase and maybe add that separately into the mix. We've not really tried any IVTT yet, so I'm not really sure how easily that would be to do, but it's something that's come up and something that we're thinking about for the future.